Hi, my name is Nathan Osterman. Today I'm going to be telling you about some of the research that I've been doing over this semester. I'm a neurology major, and this contract was from my physiology class. And so what I decided to do was look at new research that's going on in my field, and in specifically lab techniques and developments throughout history of this research. So what I did was analyze research from the University of Mainz in Germany. What they were covering was on oligodendrocyte precursor cells in mouse brains in Germany. These are a type of glial cells that characteristically express the protein NG2, which is a transmembrane protein that is expressed by this particular glial cell. And they were looking at the influence that these cells had on neuronal synapses. Now, this research is new and actually really interesting because contrary to the popular view of glial cells, which introduced by this guy in the 1850s, Rudolf Virchow defined glial cells as being passive elements of the brain that really didn't have any influence at all on neuron synapses. What they're finding out now, and in this specific research, they're looking at oligodendrocyte precursor cells, or OPCs, actually do have an effect on neuronal synapses. And they're specifically looking at the mechanisms involved. And so what they're studying was mouse brains. And so they're looking at these cells in mouse brains. And the reason that we're finding out now, as opposed to way back then, that these cells actually have effects in neural synapses is due to advances in technology, and specifically in microscopes. So previously, back in the 1850s through mid-1900s, the way that you would have drawings and stuff is they'd look through microscopes. Scientists would look through microscopes and they'd make drawings by hand. So these are some drawings made by the prominent neurologist Ramon y Cajal. You can see they're very detailed, but again, with modern microscopes, we can see we can look at 3D images in real time of like living cells as opposed to dead cells. Like this cell, they had to use stains and they could only look at it and it's kind of a 2D image. They could still derive a lot of information from that, but still, with technology that we have now, we can actually see individual protein structures and individual cells and study them in a lot more detail. And following with that, in my particular paper that I was analyzing, the use of fluorescent dyes. These were developed in the 1990s. Fluorescent dyes are used to tag specific tissues or specific proteins inside of living organisms, and they can be used sometimes as an antibody. So like you'll infuse a tissue culture with the fluorescent dyes with a solution of them, and they'll bind to their target molecules. What I found was really cool is there's also another way you can use these fluorescent dyes. You can actually genetically modify organisms to basically grow with the fluorescent dye being produced inside them, and that's how they did it in the paper from Mainz, Germany. They had, in these mice, they bred them with a red fluorescent dye to mark the neurons, as we see here, and then the green, it's actually yellow fluorescent dye, but when it shows in pictures, it comes out green. But these are the oligodendrocyte precursor cells that they're showing here, and then you can see in yellow, that's where they're overlapping and interacting with the neuron. And the second huge technological advance in lab procedures that is essential to this particular research was the development of whole cell recordings in the 1970s. This enabled us to look at individual cells. We can look at individual ion channels and individual proteins inside of a single cell wall and analyze their electrical activity and kind of their activity, like what they're doing in the cell. Whereas before we could only look at kind of the whole cell in general, and we couldn't look at specific ion channels. Now we had the ability to look in great detail at what was going on at that tiny of a level. Another procedure that they used that was really helpful in this particular research was gel electrophoresis. So they were using it to find percentages and like where the NG2 protein was kind of the main, like what they're looking at, because these OPC cells expressed the NG2, and that was these NG2 proteins are actually what was affecting in the neuronal synapses. And what they did was they would take these proteins or take solutions and they'd put them in the gel electrophoresis. So you put them into like these tiny wells inside of the gel, and then you run a current through them, an electrical current through them, and the proteins will separate out. So all the different proteins in the solution will separate out based on molecular weight, and the length of the protein. So they were able to find out exactly how many, generally how many, and where the NG2 proteins were appearing in the cell. And then Western blotting was a way that you could take the gel, put it in a solution, and transcribe basically the blot, or transcribe the gel onto a tissue so that they could analyze it better and actually analyze where these proteins were occurring and what proteins they were, because it's kind of hard because in the gel, I mean, they're inside of a gel matrix, you can't actually access the proteins very easily. And so when they blot them onto this tissue, you can actually analyze the proteins individually and see what they are and what they're doing. And so in this research, okay, so what they're looking at is long-term potentiation in the mice. Long-term potentiation is where you'll have a nerve synapse, and so you'll have the presynaptic neuron will release glutamate that'll bind to receptors and basically shoot the synapse down into the postsynaptic neuron. What they're looking at long-term potentiation is when a side effect, so you have the neuron synapse, but you also have a paracrine release, and that sends a message to the presynaptic neuron telling it to release more glutamate. And then you also have another second messenger saying to add more receptors for glutamate into the postsynaptic neuron. So basically every time you have a nerve synapse go on, it's telling it to make the synapse stronger. What they're finding out in my research is that, so the oligodendrocyte precursor cells, they have this NG2 protein, and it's transmembrane protein. So it's got pieces on inside the cell membrane and on both sides of it. What they're finding is that this section outside of the cell, so in the so you have the inside the cell, and the section outside the cell is cut off by enzymes, and it's floating around inside the synapse, basically inside the synapse, and that actually binds to these AMPA receptors, changing their shape and altering. So it basically strengthens their effect. So as receptors, so basically it enhances the long-term potentiation of the synapse. And so that's really, really interesting, especially because everybody thought glial cells didn't really do anything, but now we're finding out that these OPC cells are actually enhancing and regulating our nerve synapses inside of the neurological network, inside of the neural network. And so that was kind of that was all I was looking at was, that was there you go, sorry.